Good morning, sir.
Morning, morning.
Good morning, everyone. Welcome to today's class at Tawheed Foundation for Allied Health Sciences. Today's topic we are discussing about microscopes, different types of microscopes, and their functions.
Madam, will
Before
you share in this no slide mode?
it is not in slide mode, sir.
No, no.
1 second. Now, sir.
Yeah, please.
Good morning, everyone. Welcome to the today's class at Tawheed Foundation for Allied Healthcare Sciences, Bangalore. Today we are discussing about microscopes, different types of microscopes and their functions. Before heading into the types of microscopes, let us go into the historical timeline of microscopes. In the 1st century AD, the Romans invented the glass and used them to magnify objects. In the early 14th Italian spectacle makers. In 1590, two Dutch spectacle makers, Hans and Janssen, created the first microscope. It was a simple tube with two lenses system and had 9x magnification. In 1670, Robert Hooke, an English chemist, mathematician, physicist, and inventor, improvised the microscope of that time and developed the compound microscope. He first developed three lenses microscope. Later, in 1675, Antony van Leeuwenhoek created single lens microscope of 270x magnification. Using this microscope, he first observed the bacterial cells. In 1729, Moray Hall first presented and used achromatic lenses in the microscope. In 1830, Joseph Jackson Lister suggested the use of multiple low power lenses to achieve clear magnification. In 1878, Ernest Abbe, a German physicist and optical scientist, developed a mathematical theory relating wavelength with image resolution. So he was the first to develop and use water and oil immersion lenses. In 1903, Richard Zismondi invented ultramicroscope. This could view objectives smaller than that of wavelength of light. In 1932, Fritz invented the phase contrast microscope. In 1938, Max Knoll and Ernest Ruska invented the first electron microscope. It was transmission electron microscope. So we are going to discuss different types of microscopes later. It used a beam of an electron instead of light to make an enlarged image. In early 1940, Russian physicist Sergei developed concept of ultramicroscope, but only in 1970 it came into the working model. In 1942, Ernest Ruska improved this transmission electron microscope into scanning electron microscope. In this type, electron beams are passed across the specimen instead of passing through it as in transmission electron microscope. In 1951, Nixon and Ellis invented X-ray microscope. In 1981, Binning and Roher invented scanning tunneling microscope. This allowed us to get the 3D image of the object. So these were the early discoveries of microscope by different other scientists. Going into what is microscopy actually. Microscopy can simply be understood as the use of microscope. Microscopy is defined as the scientific discipline of using microscopes for getting magnified view of objects that can be viewed by naked eye, like bacteria, viruses, which are very minute. Those organisms are used under the microscope to visualize the structure of the organisms. It is a very important tool in biology and nanotechnology. In microbiology, it is one of the most important tools used in observing microbial cells. Medical sectors, pathology, histology, molecular biology, and cytology are in great depth of microscopy. Importance of microscope in bacteriology. Visualizing the bacteria, which is ranging from zero point 0.5 to 5 micrometers in size, which is below the resolution limit of human eye, that is approximately 20 micrometers, that can be identified under microscope. Microscopy enables the observation of these organisms, facilitating the study of morphology, structural behavior, and other anatomical characteristics. Coming to the classification of microscopes, that is divided into four types: optical microscope, electron microscope, scanning probe microscope, and X ray microscope. Let us go in detail about these classification. In optical microscope, again it is classified into bright field microscope and dark field microscope. Optical microscope is the microscopy technique that uses transmitted visible light, either natural or artificial, for developing the image of an object. So it is important and most common type of microscopy. It is further classified into bright field and dark field. Bright field microscope uses transmitted light, that is low contrast of approximately 1300 magnification, and absorbs living cells. Dark field microscopy uses reflected light. This is ideal for specimen like motility studies. This is compound microscope, which is classified under bright field microscope. Let us go in detail about compound microscope. A microscope with a high resolution and uses two sets of lens, and this eyepiece can be a single one or double eyepiece with two-dimensional image. 2D image can be observed in this one. The term compound refers to the usage of more than one lens in microscope. So also the compound microscope is one of the types of optical microscope. The other type of optical microscope is a simple microscope. The difference between simple and compound microscope is that simple microscope uses only one lens and compound microscope uses more than one lenses. The upper end of the body tube has a small fixed tube, which is known as draw tube, is to hold the ocular lenses, rack and pinion. To bring the objective screws under focus, the rack and pinion are either attached of the body tube or to the stage. These are two pairs of adjusting screws that are used either for a coarse adjustment, this one, either for the coarse adjustment, and this one, stage adjustment, for fine adjustment. When a fine adjustment is made, the body tube of the stage moves extremely short distance, while the coarse adjustment is made. The body tube and the stage move up. Through fine adjustment, you can sharp the obtained images. This is coarse adjustment, where you can adjust back and forth. This is left and right, and this is your fine adjustment. The rack and pinion have a small screw that is used for stopping the downward sliding of the body tube. This will prevent damage of the objective lens. So if you make it very close to the lens, there may be a chance that the cover slip or glass slide, which is kept for the observing, may break. So this will prevent in that one. Optical parts of microscopes are diaphragm. The amount of light falling on the object can be controlled through diaphragm. It is present below the stage. This one. The disc and iris are two types of diaphragm. Next is the condenser. Condenser, it is present below the diaphragm. The focusing of light can be done by adjusting the condenser by moving it either up or down. Next is the reflector. A reflector is a mirror that is attached above the base. One side of the mirror has the plane mirror while other side has concave mirror. When the light is strong, the plane mirror side is used, and the when light is weak, concave mirror is used. Depending upon the presence of light, it is switched into either mirror, plane mirror, or the concave mirror. The light on the object is directed with the help of reflector through the diaphragm and condenser. Objective lenses, these lenses are present over the nose piece. There are two or three types of objective lenses: low power, high power, and oil immersion. Every compound microscope with two eyepieces usually now generally comes with three eyepieces: low magnification, high magnification, and oil immersion. The objective lens is a compound lens that forms a real inverted image of the image which is kept in the body tube. Ocular lens is also known as the eyepiece. The image of microscopic objects can be viewed through these lenses, eyepieces. There are four types of magnification that can take place in ocular lens, that is 5x, 10x, 15x, and 20x. Here, objective lenses will be of 10x, 40x, and oil immersion, that is 100x. The binocular head is the device that uses two eyepieces and has many mirrors and prisms, which makes passage of light easier, and the object whichever kept in the stage can be appeared clearly. Uses of compound microscope: the identification of diseases becomes easy in pathology labs with the help of a compound microscope. Forensic laboratories use compound microscopes for detection of human fingerprints. The presence of metals can be detected with the help of a compound microscope. The study of bacteria and viruses becomes easy with the help of compound microscope. Schools also nowadays uses compound microscope for academic purposes. See the reflected light. The reflected light.
Hello. Madam, will you share that screen? Hello.
Hello. Hello, sir.
Yes, madam. I think internet issue, I think, madam.
Yeah, maybe. Is my slide visible?
Yeah, right now it's visible.
Oh. So dark field microscope, the reflected light is used instead of transmitted light to form a magnetic image. It is used to observe very thin specimens and motility of flagella and cilia. Every minute structures of bacteria can be identified in dark field microscope. So this is the working principle of dark field microscope, where the light sources come from here and the central aperture reflects the light with the help of condenser lens. The specimen which is kept here, the light that is passing through the light source will scatter, and that is observed under the objective against dark background. White cells against the dark background. Advanced optical techniques like phase contrast microscope. The phase contrast microscope is used. It converts phase shifts into differences in intensity of light, producing more contrast images. So it is used for observing living cells and cellular structures. Fluorescence microscopy. This is the phase contrast microscope. You can see the working principle of the phase contrast microscope. Light sources with the help of annular link condenser lens. The light reflects and the specimen light is scattered with the help of direct light and other lenses. The face plate is present here and the image can be observed here in contrast mode. Fluorescence microscope. It is a microscopic technique that uses a fluorescent microscope with UV light source. It is widely used in detecting antigens, antibodies, and other macromolecules. Specimen preparation will be different for each of the microscopic technique. Fluorescence microscopy specimen preparation. After preparing the specimen, it is kept here. And with the help of light source, UV light source, the excitation filter and emission filter emits the organism's light source, and the organism's images can be observed with a fluorescent color. That is usually with the help of staining. A fluorescent stain, when you apply, the cells appear fluorescent against the dark background. Confocal microscope is a newer microscopic technique that uses a focused laser beam. It is used to get high resolution 3D image of biological samples. With the help of laser, micro lenses, lens discs, and all CCD camera it is attached to it, the specimen can be observed with the help of the flow of light. Differential interference contrast microscope. It is a microscopic technique used in unstained and transparent sample to enhance the contrast of their image. Some of the samples cannot be stained with any of the stains. In such cases, this microscopy can be used to identify transparent samples and the samples which cannot take up any of the stains. Coming to the electron microscope, electron microscopy uses electron beams for high magnification. This is further classified into scanning electron microscope and transmission electron microscope. These are the two images of scanning electron microscope and transmission electron microscope and the working principle of scanning and transmission electron microscope. Let us go to the working principle of electron microscope. Basically uses signals arising from the interaction of electron beam with the sample to obtain information about structure, morphology and composition. The electron gun generates electrons. Two sets of condenser lenses focus the electron beam on the specimen and then into the thin tight beam. To move these electrons down the column, an accelerating voltage, mostly between 100 to 1000 kilovolts, is applied between the tungsten filament and anode. The specimen to be examined is made extremely thin, at least 200 times thinner than those used in optical microscope. In compound microscope, you are preparing on slide, whereas in transmission electron microscope or scanning electron microscope, specimen needs to be prepared in a very thin glass film, like your cover slips. It has to be prepared on that. Ultra thin sections of 20 to 100 nanometers are cut, which is already placed on the specimen holder. The electronic beam passes through the specimen and electrons are scattered depending upon thickness of refractive index of different parts of the specimen. The denser regions in the specimen scatter more electrons and therefore appear darker in the image. Since fewer electrons strike the area of the screen. In contrast, transparent regions are brighter. So the electron beam coming out of the specimen passes to the objective lens, which has high power and forms the intermediate magnified images. The ocular lenses then produce the final further image, which is connected to the monitor, and the image can be observed there. Coming to the transmission electron microscope, particularly, this transmission electron microscope is used to view thin specimens through which electrons can pass, generating a projection image. The transmission electron microscope is analog in many ways to conventional compound microscope. It is used, among other things, to image the interior of cells. Thin sections of the cells can be made, and the structural characterization of the cells can be identified by transmission electron microscope. The structure of protein molecule can also be observed. The organization of molecules in viruses and cytoskeletal filaments can be observed, and the arrangement of protein molecules in cell membranes can also be identified. Very minute structures of each and every cell can be identified by transmission electron microscope, since electron ions are participate in reflecting the image. Scanning electron microscope is conventional scanning electron microscope depends on emission of secondary electrons from the surface of specimen. So, because of its great depth of focus, a scanning electron microscope is analog of stereo light microscope. It provides detailed images of the surface of cells and whole organisms. Scanning electron microscope is used to detect abnormal structures in cells and organisms. A scanning electron microscope because the image is focused by scanning electron beam onto the surface of specimen in a faster pattern. Applications of this electron microscopes are used to investigate ultrastructure of a wide range of biological and inorganic specimens like microscopic cells, large molecules, biopsy samples, metals, crystals can be identified in electron microscopes, which are usually not possible in bright field or any other different types of optical microscopes. Industrially, electron microscopes are often used for quality control and failure analysis. Modern electron microscopes produce electron micrographs using specialized digital cameras and frame grabbers to capture the images. The science of biology owes its development to electron microscope. The study of microorganisms like bacteria, virus, and other pathogens have made the treatment of disease very effective. This is scanning probe microscope SPM uses the technique of a physical probe to scan specimens and form magnified images. This measures the surface features of the specimen. This is the working principle of scanning tunnel microscopy, atomic force microscopy, scanning near field optical microscopy. So there are three different types of scanning probe microscopy. X-ray microscopy is a microscopic technique that uses soft X-ray radiation to produce magnified image of the specimen. The condenser optic, when the sample is kept here, soft X-ray radiations are used to reflect the light and the images are being observed here. This is the working principle. There are some of the key terms which are used in microscopic technique, that is magnification, magnifying power, resolution. Magnification is the process of producing an enlarged image of specimen by using a lens system. In a microscope, magnification can be computed by calculating the product of magnification power of the eyepiece by the magnification power of the objective in use. So total magnification can be calculated, that is power of the eyepiece into objective lens. Magnifying power is defined as ratio of angle subtended by the image at the eye to the angle subtended by the object at the eye when placed at a minimum distance of distinct vision. It can be mathematically defined as m is equal to 1 plus dfm, that is equal to 1 plus by fraction d. M refers to magnifying power, d refers to least distance of distinct vision, f refers to focal length of convex lens. Refractive index and resolution. Refractive index can be defined as velocity of light in a vacuum to velocity of light in a medium, that is in a substance. Simply, it is measure of bending of a light ray when passing from one medium to another. Mathematically, it can be defined in this formula. Resolution can be defined as the shortest distance between 2 points on the specimen that can be distinguished by a microscope in its image. It is the ability of microscope to distinguish detailed on the specimen. Mathematically, wavelength is calculated by pi to NAR, where pi is wavelength, R is resolution, and Na is numerical aperture. Microscopy revolutionized science and medicine from simple lenses to electron beams. It continues to evolve with nanotechnology and imaging annotations. So, 3 marks questions can be of phase contrast microscope. Explain phase contrast microscope. You can explain I'll just show you, working principle of phase contrast microscope, how the light sources come, how the lenses go inside when the specimen reaches the light source, how it is directed to form the image. That image can be explained for phase contrast microscope. Then 5 marks questions include briefly explain the parts of compound microscope with need diagram. Explain the part of microscope with need diagram is this one, with all the labeling part: stage adjustment, eyepiece, no piece, objective lenses, stage, condenser light. This is, and electron microscope can also be asked for 5 marks question, where you have to explain what is electron microscope, scanning and transmission electron microscope based on the working principle you can explain, and also the advantages, disadvantages, and uses of scanning electron microscope can be explained. So this is about microscopy, types of microscopes, and the questions which is covered in under the microscope are covered in this chapter.
Thank you, madam. Thank you so much. Very
Thank you,
nicely you
sir.
have covered. It was excellent presentation, and
Thank you,
most
sir.
of the things are covered in this microscopy.
Thank you, sir.
So next time, what we'll do, we will ask our students to join, madam. So if
Okay,
you
sir.
have any questions, let
Mm.
them answer. Let them
Oh,
raise the queries there itself in
okay, sir.
Q&A session. Okay.
Okay, sir.
Yes, madam. So we will have one more PPT. We'll end up with this.
I have prepared for one more PPT. We will
Yeah.
go for morphology of bacterial
Done,
cell.
done.
Is my slide visible?
No, no. Yeah.
The next topic we are going to discuss about is the morphology of bacterial cell. Bacteria are a type of biological cell that is prokaryotic and unicellular. Due to the lack of membrane bound nucleus, these are simpler than other types of living organisms. Although only some of them can be seen by naked eye, while the rest are microscopic, they display a wide range of shapes, size, and structure. Bacterial size is the unit of measurement used in bacteriology, that is in microns, which is 1,000th of the millimeter. Bacteria are in general 1/10 the size of eukaryotic cell. So overall size of the bacteria ranges from zero point 0.5 to 5 micrometer. However, they can be as tiny as zero point 0.3 micrometers and as large as zero point 0.7 mm. The limit of resolution with the unaided eye is about 200 micron, and as many bacteria are smaller than this size, they are not visible with naked eye. So microscopes has to be used to identify bacteria. The size of common bacteria like Escherichia coli ranges in size from 1.1 to 1.5 micrometer in diameter. It has been observed that the size of bacteria has a significant role in the survival of organisms. Owing to their tiny size, they are capable of surviving and even thriving in various unlikely environments, like vertical sediments in the marine environment. Since other organisms are absent in such environment, bacteria utilizes the available resources. So, beside the small size of bacteria favors parasitism and the ability to survive in areas with low nutrition, high surface area volume ratio allows the bacteria to take up nutrients required for survival while steady growth and reproduction. Importance of small size, they thrive in extreme environment. They have high surface area to volume ratio, efficient nutrient uptake, favor parasitism and survival in low nutrient areas. Coming to the shape of bacteria, most of the bacteria have a rigid cell wall that provides a different shape to the bacteria. This is because of the cell wall that gives the shape to the bacteria. Even though this characteristic is valid for majority of bacteria, they vary in shape that allows them to classify and identify easily. This wide variety of shape is determined by bacterial cell wall and cytoskeleton. Even though bacteria have a wide variety of shape, any genus typically exhibits a limited subset of morphologies, indicating that with the universe of shapes to choose from, individual bacteria adopt only those that are adaptive. Bacteria with different shapes present different physical features, and these features help cells cope up with external environment. It has been observed that bacterial shape contributes a measure of survival valve in face of nutrient acquisition, cell division, attachment to surface, and other external differentiation. The common shapes of bacteria are cocci, that is round single spherical shapes. These bacteria may either remain single or it can be attached to one another. It can be attached in chains. We'll just go into the arrangement of cocci. It is assumed that coccoid forms were derived from rod shaped organisms through evolutionary time. So, some of the examples are Streptococcus, Staphylococci, Enterococci. These are the arrangement of cocci. Coccus, single spherical cell, diplococci, pairs of cell that can be Neisseria gonorrhoeae, Streptococcus pneumonia. Tetrad are four. I'll just show you how the tetrad and other things are there. See, this is monococci, diplococci, tetracoccus, four cells, diplo two cells, sarcina, group of cells, usually eight, streptococcus, chain of cells, staphylococci, cluster of cells. So tetrad are group of cells, pediococcus, eriococcus, sarcina, cube of eight cells, sarcina lutea, streptococci is chains of cell, Streptococcus pyogenes, Staphylococci, group of cells that is Staphylococcus aureus and any of the Staphylococcus species come under this arrangement. Next is bacilli, usually called as rod-shaped. These are rod-shaped cells that also, like cocci, remain either single or attached to one another. Bacilli cell bacteria are among the first bacteria to have arisen, and this shape is said to be not as advantageous as other shapes. This has been assumed upon the observation of behavior of E. coli cells, which, though motile and chemotic, move slowly and cannot tumble to change direction like cocci. Example arrangements of bacilli cell, single cell, if it is single rod shaped, it is Bacillus cereus. If it is two rod shaped cells, that is pair of rods, Klebsiella. Streptobacilli, chain of rod cell, Streptobacilli moniliformis. Cocobacilli, Cocobacilli is a form of cell where it appears both as cocci and bacilli. See here. Round shaped single, rectangular, plum that is bacilli, accumulative, club shaped, curved. So there are different types of bacilli cell. Coco bacilli is Haemophilus influenza. Usually either it is cocci or bacilli. Once when it is observed, it appears as a cocci, and when you observe, it appears as bacilli also. So when it has a dilemmatic structure, it is called as Coccobacilli. Palisade arrangement like Corynebacterium, diphtheria. Spiral shaped organism. This group includes bacteria that are either helical shaped or curved, comma shaped bacteria. It ranges from slightly curved to corkscrew like. And let us go into arrangement of this organism. Vibrio is comma shaped, Vibrio cholerae. Spirochetes, which have flexible spirals with axial filaments. Treponema pallidum is an example. Spirille is rigid crock shaped like Helicobacter pylori. You can see the differences of organisms here, spirochetes and other. Apart from this cocci, bacilli, spirals, there are other shapes and arrangements which cannot be characterized in any of these classification. Those are appendaged bacteria, the bacteria that produce a unique structure like pilus or fimbriae. In future classes, when we come into the structure of bacterial cell, I'll go into the pilus and fimbriae. These bacteria are more virulent than other bacteria that do not form these appendages, like Neisseria gonorrhoeae, which causes gonorrhea infection. Box shaped or rectangular bacteria, these are rectangular in shape and resembles a box. Club shaped rod bacteria, these bacteria are thinner on one side than the other. One of the classic example is Corynebacterium of this club-shaped bacteria. Filamentous bacteria, these are bacteria that are long, thin, and filament shaped. They sometimes divide to form branches resembling strands of hair, these are called as mycelium. Example of this is actinomycetes. Triangular shaped bacteria. This group includes bacteria that are triangular in shape like Haloarcula. Pleomorphic bacteria, these bacteria that do not have any specified shape, include with so this cannot be included in any of the group. They can change their shape, but in pure culture, they appear to have a definite form, like Mycoplasma pneumoniae, Mycoplasma genitalium, Rhizobium also for that matter. Stacked bacteria, these are the bacteria that possess stack on one another of the cells, like Colobacterium. Star-shaped bacteria that look appears like a star, like Stella humosa. Bacteria vary widely in size and shape. Morphology influences survival, motility, and adaptability. So, classification based on shape and arrangement aids identification. Basically, these shapes of the bacteria is very important to identify under which genus it belongs to and also to study the characterization of the bacteria. Questions that arise from this topic is explain the types of cocci. For this, you have to go. Cocci is a spherical shape organism. You have to explain about the characteristics of cocci, arrangements of cocci like monococcus, diplococcus, tetracoccus, that is of four. Tetracoccus is also called as tetrad, four cells arrangement, sarcina, streptococci, and staphylococci, that is group of cells with an example. Then write about bacterial size. Bacteria is very minute, which ranges from zero point 0.5 to 5 micrometer, and those size related things. 5 marks is explain the arrangement of bacilli with examples. Monobacilli, diplobacilli, streptobacilli, cocobacilli with examples, arrangements that can be shared. So this is all about the arrangement of cells, morphology of cells. In next class, we'll go into the structure of bacterial cell, where we are discussing about the cell membrane, plasma membrane, pili fimbriae, flagella, endospores. All the internal structures of bacteria will be discussed in the future class. This is a general introduction to the morphology of bacterial cell, which have different shapes of bacteria.
Thank you, madam. Thank you. It's a wonderful class.
Thank you, sir.
Thank you. Shall we wind up, madam?
Yes, sir.
Thank you.